Good evening, everyone. Please confirm if you all can hear me. Is the screen visible? Please confirm. Good evening, Hemlata. Okay, thank you for confirming. So I hope everyone has joined. So welcome to another interesting session of Brahmastra Series. So as you all know that I have been discussing the questions from Unit 8. So I have been discussing questions subunit wise from Unit 8. But as discussed in previous session and today also, I will be discussing the mixed questions. The questions based on the topics from subunit 8A to 8E. So unit 8 is all about inheritance biology. And so far we have discussed different variety of questions that has been asked in CSIR NET exam. So I'm sure you get an idea that what type of questions are expected and how you have to solve them. I have also discussed the strategies and the tricks to solve the questions. So some more questions we'll be discussing today. And the questions, like it will be mixed questions as you get in CSIR NET exam, right? You don't know that from which topic the question will be asked, right? So far we have been discussing particularly questions on probability or question on linkage or question on physical mapping. So you know that today's session is all about the questions based on a particular topic. But today I will be giving you mixed questions for solving, for practicing. So I hope you all are excited. Yes? Are you all excited to practice questions from Unit 8? Can I get some responses in the chat? So I hope I'm audible to everyone. Okay. Thank you, Hemlata. Thank you, Aishwarya. Thank you for the response. Thank you, Anu. Thank you, Suresh. Thank you, Surendra. I can see some of the participants, they are attending all the sessions. I really appreciate you all. I'm sure that definitely these sessions will definitely going to help you. And I'm sure you yourself are seeing the changes. Like when you're practicing the questions, I hope now you are doing it faster. You are recalling the concepts much faster and are able to solve the questions much faster. Yeah, very good evening, Anusha. Good evening, Deepshika. Welcome to the another session of Brahma series. So let's get started. Let's start with the first question. So here comes the first question on your screen. The question is a plant of the genotype, and the genotype of the plant is mentioned. It is self. Now, two genes are linked and are 50 map units apart. What proportion of the progeny will have the genotype which is mentioned? That is double homozygous recessive condition. So I'm giving you some time to solve. Please solve it and tell me what is the answer here. So as I always say, look for what information is given to you in the question. You can write it down separately also. Like initially when you are practicing the questions, you can do it that way. So you can know that you never make a mistake in missing any kind of information given in the question. So this is a small question, so it's fine. But specifically when there are the questions which are very large, like a lot of text is there, then definitely this kind of strategy you can use. So the answers I'm getting is option 4. Suresh, Surendra, you have already answered. Aishwarya, okay, you are also saying option 4, that is 1/16. So let's others also solve and then I'll let you know what is the correct answer here. Okay, Anupam, you are also saying option 4, 1/16. So here you need to recall what is linkage, what is the meaning of 50 map units here, and what is the proportion of the progeny with this genotype. Okay, Anu, you are also saying option 4, Anusha, option 4. So far I'm getting a uniform answer, so that's great. I hope everybody who is attending the session is solving. Please take your time. I'm giving you some time to solve. At least try. You need not to devote extra time for solving the questions in the class itself, in this session itself. You can solve the questions, right? We all can solve it together. Okay. So as I told you that you need to understand what is linkage here and what is the meaning of 50 map units here. So as I discussed in the session, Brahma series session, where I discussed the questions on linkage, I told you about this particular flow chart, right? So it's time for you all to recall this flow chart. I told you that the linkage, it's a deviation from expectations based on Mendelian principle of independent assortment. And how will you identify that it's a case of linkage or it's a case of independent assortment? So linkage is where the genes are located on the same chromosomes and they are always inherited together. So they are located on same chromosome, closely located, and they are always inherited together. So we call them as linked genes. And the linkage can be of two types. It can be complete linkage or incomplete linkage. So in complete linkage, no crossing over happens. And the gametes form will always be parental type. But in case of incomplete linkage, the crossing over can happen because linkage is rarely complete. And even in independent assortment, like in Mendel's dihybrid cross, we have seen that the new recombinants are produced, right? So new recombinants formed is because of crossing over only, right? So in case of incomplete linkage and in case of independent assortment, crossing over can happen. But the difference is that in case of incomplete linkage, the gametes formed are the parental type more in number and recombinant type will be less. However, in case of independent assortment, the parental type and recombinant type will be in equal proportions. So basically, to test for linkage, we perform a test cross where we perform a cross between a F1 or the heterozygous with that of homozygous recessive parent. So as mentioned in the question that the gene A and B, they are linked and they are 50 map units apart. So basically, I'm sure you know that in genetic mapping, we have discussed that the genetic maps, they are plotted, they are calculated on based of recombination frequencies. The distance between the genes are calculated on based of recombination frequency. And the unit that we use is MU, map unit, or we can also use centimorgan. And 50 map unit here, it means 50% recombination. So distance between the genes is 50 map unit, or I can say that 50% recombination, the frequency of recombination is 50%. So if you are performing a test cross where we perform a cross of a double heterozygous with that of homozygous recessive parent, so you all very well know that the gametes formed by the double homozygous recessive parent will be of one type only, right? But here, the double heterozygous parent can form the four type of gametes, right? Where dominant alleles coming together, or we have the dominant allele of one gene and recessive allele of another gene, or vice versa, or maybe both the recessive alleles are coming together, right? So different gametes will be formed where you can see that two will be like if you see the proportion, so it is always in ratio of 1 is to 1 is to 1 is to 1, right? So we can say that 50%, like when you're performing a crossing with a double heterozygous with that of homozygous recessive parent, it will result in 50% recombination and 50% parental gametes. Now, this 1 is to 1 is to 1 is to 1, that is equal proportion, is always seen in case of independent assortment and not in case of incomplete linkage. So one thing is very much clear that here the genes, they segregate by independent assortment as proposed by Mendel. You can do it directly also. You know that, okay, 50% is the recombination, it means 50% will be the parental, right? And if you talk in terms of gametes, so like 25%, 25%, 25%, 25%, each four gametes with equal proportion will be there, right? So genes here segregate by independent assortment as proposed by Mendel. And when you perform the selfing of this double heterozygous, which is mentioned in the question, so it's like a Mendel's dihybrid cross, right? And you very well know that in case of Mendel's dihybrid cross, when you talk about the genotype, that is double homozygous recessive, the proportion or the proportion of the progeny with this genotype is always 1 by 16. So if you have a knowledge about the linkage, about the independent assortment, about the dihybrid cross, you can easily answer this question. So dihybrid cross, where we consider the parents that differ in two genes. So here also we have the two genes A and B, alleles of two genes A and B. And on performing the selfing of the double heterozygous, it results into 1 by 16 because it's a case of independent assortment. And because the here 1 by 16 is the answer, so the correct option is option 4. So I'm glad to see the responses. Sunshine, very good. You have also marked option 4. Moving over to the next question, that how many linkage groups are present in human males? Now this question is again very direct question, and it is based on a concept of linkage group. So if you know what is a linkage group, you can easily answer this question. So these type of questions are usually asked in part B of the exam, right? Part B in the exam. So quickly answer this question. I don't think you require more time to solve this question. So you have to keep a track of the time also. So let's see what is the answer you are choosing here. Okay, Achal, Aishwarya. These questions I have given in the weekly test and quest, right? So I hope there you have also solved these questions. Okay, Achal, Aishwarya, Anusha, you are saying option 2. Fine. Okay, Saujanya, Anupam, you are also saying option 2. That is 24. Sunshine, you are also saying option 2. Okay, so again, I'm getting a uniform answer. So that's great. But let's see if it is correct or not. I'll confirm it in a minute. Please try, everyone. So here, what is the meaning of linkage group, basically? So you need to understand that all the genes which are located on one chromosome, they are considered as a linkage group. Okay, like for example, if you see here, this is gene 1. And here, if you see, this is gene 2, this is gene 3 and gene 4, right? So they forms a linkage group. So all the genes present on one chromosome are considered as linkage group. And you can also say, also define it as a set of genes at different loci, that is at different location on the same chromosome, that except for crossing over tend to act as a single pair of genes in meiosis, and instead of undergoing an independent assortment. So basically, they all are inherited together, right? So as I told you that except for crossing over, they tend to act as a single pair of genes in meiosis instead of undergoing independent assortment. So in a very easy way, you can remember that all the genes present on one chromosomes are considered as one linkage group. So number of linkage groups of a species corresponds to total number of different chromosomes of that species. Now, important thing to note here is, kindly note that it is not simply the haploid number of chromosomes in haploid cell. Okay, so this point you have to keep in mind. Now, if we take the examples of human, so we know that in males we have 22 autosomes, and along with it we have X and Y chromosome. But in female we have 22 autosomes and we have two X chromosomes, right? So males have 22 pair of autosomes, one X chromosome and one Y chromosome. So 22 pair of autosomes have like this is the 20 pair of autosomes. So the chromosomes, each chromosome will be having many genes. And this X and Y chromosomes are not same. they are also different, right? There are different genes present on X chromosome and Y chromosome. So that is why here the total linkage group will be 24. But if it has been asked about the female, then there are 22 pair of autosomes, but as 2X chromosomes are there, so total linkage group will be 23 because genes located on X chromosome will be same. So therefore, the correct answer here is option 2. So you all have answered correctly. Very good. Shri Vidya, very good evening. I have discussed 2 questions so far. So please join in solving the questions. Now let's move over to little bit harder questions, right? Specifically when it comes to part C, where you have to score 4 marks, right? So each question carries 4 marks. So there you can expect such type of question. So by looking at the question, you can easily identify that it's a question on gene mapping, right? And particularly, I have told you earlier as well that the gene mapping is very important from exam point of view. Topic gene mapping is very important from exam point of view. So here you can see one such question. Please go through it and try to solve it. So for this, you will be requiring some time. So I will be giving you few minutes to solve. So in Drosophila, bar I is a dominant mutation, while miniature wing and yellow body color are recessive mutations. Heterozygous females for these mutations were crossed to normal-eyed miniatured winged and yellow body colored males. Now this is the progeny which was obtained. Based on the results obtained, you have to find out the order of the genes. And even they have mentioned the order, like is it BMY or is it MBY? And also you have to find out the genetic distance between B and Y allele. So is it 40 centimorgan or is it 17.1 centimorgan? So you have to find out the correct combination of the answer. So I hope you remember I have given you a flow chart for this as well. I have given you the sequence of steps. So I told you how to determine the, like what is the criteria for determining the gene order, what points you have to keep in mind while determining the order of the genes, and how you can calculate the distance between the 2 genes, how will you calculate the recombination frequency. So it's a step-by-step process. So you just have to recall that and you have to try answering this question. So if you find out the order of the genes, you can omit 2 options out of 4 because order will be either A or B. So if you are saying A, then that means answer cannot be 2 and 4. But if you say that the B gene order, the gene order given in option B is correct, then in that case option 1 and 3 will not be correct. So this way you can omit 2 options out of 4. Okay, hello Aishwarya. Sure, I'll explain you, Aishwarya. Let's everybody solve and then I'll explain you. And I think you have missed the class, so you can go through the class recording as well. Okay, the details are there. And in case of any doubt, you can then ask me during the class or at the support. But even now also, I will be discussing. Okay, so let's everybody solve and then I will discuss the flow chart with you. So yes, what do you think is the correct answer here? Please quickly give me the answer. See, such type of questions, they appear to be very complex, but believe me, they are very easy. If you just remember the strategy of solving the question, the tricks wherever required, you can easily solve the question. So here is a data table given to you. Some numbers of the progeny with these genotypes are mentioned. Okay, Sunshine, you are saying gene order is MBY. Fine. So now let's now calculate the distance between B and Y. Now, if you are saying MBY, so definitely answer cannot be 1 and 3 according to you, right? So this way you can omit 2 option out of 4. So let me just quickly recall you that what steps you need to follow in case you don't remember it right now. So Aishwarya, you can also pay attention to this flow chart. So see, the very first step is if some data table is given to you for such questions on gene mapping where you have to determine the gene order or where you have to determine the distance between the genes. The very first thing is you need to identify the parental combination and the double crossover. Now, so basically when you talk about the 3-point cross, okay, so kind of a 3-point cross, that is you are considering the alleles at 3 locus, right? So basically there are 4 possibilities which can happen. So first possibility is if there is no crossing over, okay, no crossing over. If there is a single crossing over event, like for example, if you have a gene A, B, and C, right? So we can have the 4 possibilities that either the no crossing over will happen between the genes, or the single crossing over, that is where the crossing over occurs between the gene A and B. Third is single crossover event 2. Okay, so this is single crossover event 1. This is single crossover event 2, where crossing over happens between gene B and C. And fourth is where you talk about double crossover. That is crossing over is happening between both A and B as well as between B and C. So here, instead of giving this example, so this particular example I have discussed in the class in detail. And so here, instead of giving A, B, C, they have mentioned B, M, and Y, right? The 3 alleles they have. The genes, the alleles of 3 genes they have given. So very first thing is you have to identify the parental and double crossover. So from the data table which is given to you, always remember the most common ones are the parental combinations, that is non-recombinant, and the least common will be the double crossovers. So this is the flow chart you have to follow, that identify double crossover and parental most common, double crossover least common. You can compare the double crossovers and parental to find middle gene. So any one parental and any one double crossover, you can compare to find the middle gene or to find the correct gene order. Once you find the gene order, you have to rearrange the sequences in the given table. Okay, so whatever is the data table given to you, you have to rearrange the sequences. And then finally, you have to find out the single crossover events. So like here, particularly in the question, it is asked the distance between B and Y. So only one single crossover event you have to, recombination frequency you have to calculate it for. So you have to find out in the data table which are the single crossover events 1 and 2. You have to calculate the recombination frequency. So formula for recombination frequency is number of recombinants by total progeny 200. And finally, you can plot the map with the frequencies. So here in this particular question, we don't have to plot the map. It's just that we have to find out the gene order and we have to find out the distance between the gene B and Y. So from the data table, as I mentioned in the previous slide, the most common ones will be the parental. So definitely here you can see the most common ones are 165 and 185. So that means these are the parental combination. And the least number you can see here is 20, right? So that means this must be the double crossover. So let's say I take this parental combination and this double crossover. So you know very well that if I write down the BM plus Y plus, okay, so I'm writing the genotype of the parental. So this is one parental and we have, this is another parental and this is the double crossover, one of the double crossover. And so I'm comparing one parental with double crossover. So here I'm writing B, M, and Y. So if you see here, I have given you a trick that you have to always find odd one out, right? So how will you find odd one out? So you have to compare the alleles, right? So like you have to see what is the pattern which is followed. Like here, if you see, we have the recessive allele for the B gene. Okay, because wild type is represented with B plus, right? So here it's the recessive allele. So recessive allele of B gene is present on both the chromosome. Here, if you see, we have the dominant, the wild type allele for the M, but here we have the recessive or mutant allele. Similarly, here we have the wild type allele for Y, but we have the recessive allele for the Y gene. So what is the pattern? That on one chromosome there should be wild type, another chromosome there should be mutant type of the same gene, right? But here this is not, the B allele is not following that. On both the chromosome it's the recessive allele or mutant allele. So we can say that the B is odd one out. And if B is odd one out, that means this is the allele which will be in the center. Because if you relate it with the double crossover, it's always in the double crossover, the middle gene changes its position. So this is the reason the B allele must be in the center. So that means the correct gene order here will be MBY. Now I know, like if you see here, I have marked this as parental. So most common, I think there is no confusion over here. Least common 20, so that is also fine. Now I know majority of you have chosen 25 because this is the next least common, right? But please note here, the another double crossover will be 30 here and not 25. So if you have marked 25 as double crossover, this is where you are making a mistake. Now, let me explain you why here the 30 will be the double crossover. So always in case of any doubt, you can always, like very first thing that you have to see here is that if you compare the alleles, they are not given in the cis configuration. Now here you have to recall what is cis configuration and trans, like coupling and repulsion, if you remember. So coupling is where you have the wild type alleles of the genes present on same chromosome and recessive on another. Whereas repulsion is where wild type gene of one, like the wild type allele of one gene is present on one chromosome and you can see the wild type allele of the another gene is present on separate chromosome. So they are opposite. So it is trans or repulsion heterozygote. Trans configuration or repulsion heterozygote. So here also, if you see, this is the parental and this is the parental. So if I write it down, the combination, so now we know the gene order is MBY, so we can write it in correct order. So you all can write it in correct order. Please write it in your copies. So basically it will be M plus B and Y plus, right? So this is the parental combination I'm writing. And this is another combination. So that is M, B plus Y. So you can see in comparison to, like if you see here M plus, right? And the B. So here this is the wild type allele of one gene, but this is the mutant allele of another gene. So trans configuration it is. Now if the double crossover happens, so you know that in the double crossover the middle gene will change its location. So B plus will move over here and B will move over here, right? So basically you will get M plus, you will get B plus, and you will get Y plus, right? And on another chromosome you will get M, B and Y. So that is what the genotype you can see here. If I write it in correct order, it will be M plus, B plus, Y plus, which is over here. And another double crossover will be M, B and Y, which we are getting over here. So that means you can even cross check in the exam if you are getting a doubt. Okay, specifically in case of if the repulsion heterozygote is given, you can cross check. And here you can easily see that on performing the double crossover, like when the parents we are writing the genotype and they undergo double crossover. So these genotypes will get, which is mentioned, one over here and another over here. So that means these two are double crossovers. Now, as we have determined the gene order, that is which is given in option B, so that means answer cannot be option 1 and option 3. So that means we have omitted 2 options out of 4. Now you have to find out the distance, right? So once we determine the gene order, I told you, you have to rewrite, you have to rearrange the sequences. So this is the rearranged sequences. Okay, I have written it in correct gene order. Now to find the distance between gene B and Y, you have to calculate the distance is calculated based on recombination, right? Can be calculated using recombination. By calculating recombination frequencies, we can find out the distance. So B and Y, first of all, you have to identify that what is the single crossover event which is telling you that which is indicating the progeny for the for for the single crossover events, the progeny which has undergone the single crossover events. So basically here you can see again, let me write the parental combination. It is M plus B and Y plus, and another is M B plus Y plus. So single crossover event means that what is the like the crossing over between B and Y. So if the crossing over is happening between B and Y, this allele will move here and this allele will move over here. So the new combination that you will get will be M plus B Y, and another you will get is the M B plus Y plus. So now you have to look for where you are getting this combination, because see, you are left with 25, 120, 110 and 45. So there are two single crossover events. One will be crossing over between M and B allele, and another is B and Y allele. So specifically, we have to find out for B and Y. So if you look here, this is M B plus Y plus, and this is another combination M plus BY, which is shown over here. So that means these are the progenies which are getting this genotype because of the single crossover event between the allele B and Y. Now, to calculate the distance, you have to use a formula, that is number of recombinants by total progeny into 100. And you know that when you are writing the number of recombinants, so along with 120 and 110, because these are recombinants, because of we are getting because of crossing over, you have to include double crossovers also, right? Because one of the event in double crossover is single crossover. So 120 plus 110 plus 30 plus 20 divided by total progeny, which is 700 in this case, into 100 gives you 40 centimorgan. And once you get 40 centimorgan, that means answer here is option 2. So Saujanya, I hope it is clear why you will not take 25 and you will going to take 30. And I hope the answer is clear to everybody. So now let's move over to another tricky question. So please go through this question and tell me the answer. Curled wing, ebony body color, and sepia eye are three recessive mutations that occur in fruit flies. The loci for these mutations have been mapped and they are separated by the following hypothetical map distances. So map is given to you. The interference is mentioned. The interference between the genes is 0.4. Now a mutant CUESC fly, that is for curled wing, ebony body color, and sepia eye, they are crossed with a homozygous wild type fly. The resulting F1 females were test crossed that produce 18,000 progeny. So what number of flies in each phenotype class is likely to be obtained in progeny of the test cross? So 4 options are given to you and you have to answer, you have to mark the correct answer. So now here, instead of giving you a data table, they have given you some values for interference and they have given you some information that total progeny is 18,000. And based on this, you have to determine, like instead of giving the numbers directly, you have to determine the number of flies in each phenotype class. So you can see the questions on gene mapping can be asked in different ways. So here is one such question. Please go through it. So to solve this question, you need to recall what is interference, what is coefficient of coincidence. So this is these are few things that you have to keep in mind. So quickly answer these questions more quickly. Quickly you give me the answer, more questions I can solve. Yes. I hope you have understood the question. I have given this question in the test also. So this question is not very direct. So you have to solve it by keeping in mind the interference and coefficient of coincidence. So if you forgot about interference and coefficient of coincidence, so here you can recall. So these topics I have discussed in detail in the classes, regular classes. So what is coefficient of coincidence? It is the ratio of observed double crossovers to expected double crossovers. So observed double crossovers is that we usually see like in the data table which is being given in the question. So there the observed double crossovers are mentioned. As we have seen in the previous question, it was 30 and 20 which was given. And the expected double crossovers. How will you calculate the expected double crossovers? So basically expected double crossovers, theoretically we can obtain the number of expected double crossovers by multiplying the product of two single recombinant probabilities by total number of progeny in the cross. So here basically we'll be using the product rule and we'll be multiplying the individual probability of the single crossover event. So expected double crossovers you can calculate by multiplying the individual probabilities of single crossover events by total progeny. And double observed double crossovers I told you, which is the values which are given in the data table usually. But here in this question, no such data table is given to us. And what is interference? Interference is the degree to which one crossover interferes with additional crossovers in the same region. Like if you see the double crossovers are least common. So what is the reason for being least common? The reason is that maybe because of some interference or you can say steric hindrance, if the crossing over is happening here, it will not allow the crossing over to happen in the nearby region. So this is the reason that double crossovers are seen are less common. And what is interference? It is the degree to which one crossover interferes with the additional crossovers in the same region. And it is given by the formula 1 minus coefficient of coincidence. So now I have already told you about coefficient of coincidence and interference. So quickly answer this question. Okay, so Anupam, for so you can solve it right now. Okay, you can practice it right now. Some questions, see in the test and quest, we can give you few questions, and during this 1 hour session, we can solve some more extra questions, right? So the motive is that we need to practice, and more we practice, more we learn, right? So I'm definitely like why Biotechnica has come up with this Brahma series, just to help you all, because we get a lot of questions that uh, like at the end in the exam you have to solve the questions, right? And many of you asked us that, ma'am, where we can get questions for practice. So I usually give you some questions in the class, during the class, in the test, and even we'll make you practice questions like through the test or during the class, right? But other than that, even in this particular series also, you can practice the questions, right, during this 1 hour session. So go through the question. You already know the question, and I asked you all to recall about coefficient of coincidence and interference. So let's see. So here in these sessions, you also get an idea that what all concepts are important, right? In Brahma series, you can see that we are discussing the very important questions, the questions asked in CSIR NET exam, right? So even you get an idea that what all points, what all concepts you need to revise frequently as the questions are asked on these concepts, right? Are based on these concepts. So to know more about the concepts and to like if you have not associated with us, so those students who have not associated with us and they need help in preparing for CSIR NET exam or other exams, so there's an opportunity for you. You can join our Watson Junoon Scholarship. So with this scholarship, you'll get 1 year coaching for CSIR NET, GATE exam, and even if you are preparing for other exams like QAT, IIT, GEM, and GAT-B, so you'll get the the coaching here with this Watson Junoon Scholarship. Along with it, along with the coaching, 1 year coaching, you'll get 12 months of access to global and national workshops, which are conducted by Biotechnica. Also, you're going to you get access to our programming internship, and you can also get access to 4 certification courses of your choice, and also you get direct access to the coaching experts on WhatsApp, and even you get a pass to our CRISPR conference. So for more details, you can call us on our toll-free number. So number is on the screen. You can note it and apply for the scholarship as soon as possible. Even those who have already having the scholarship, they can upgrade their scholarship to this Watson Junoon Scholarship. So quickly answer the question. Okay, Sunshine, you want to see the question again. So here is the question. Please go through it. And then I'll discuss this question. Okay, Tejaswani, so please note that there are many students who are still associating with us, right? So don't worry about the topics which are already being done. It will be done for you again in the class, okay? So basically, you need not to worry about that. Like if any particular topic is missed, even as you know, we provide access to the recordings. You can go through them, right? And as the new students keep on associating with us, we need not to worry about it. We will be discussing. It will never happen that that particular topic will not be done for you all. Okay, it will be done, but like we have to go with the flow. So once the already the topics which are ongoing, once they are completed, we'll be taking these topics. But meanwhile, if you want to go through the topic, the recordings are there in the portal, which you can always go through. Okay, so let's solve the question now. So basically, interference, I told you, it is given by 1 minus coefficient of coincidence, right? And the interference value given in the question is 0.4. Now, coefficient of coincidence we can calculate, right? If you know interference, the formula is 1 minus coefficient of coincidence. If you know the interference, that is 0.4, and coefficient of coincidence you can easily calculate taking it to the other side. So it will be 1 minus 0.4, and that gives 0.6. So now you get a value of coefficient of coincidence also. Now in the question, the distance between the alleles are mentioned. So the distance between these two is 20 centimorgan and distance between these two is 12 centimorgan, right? Now basically coefficient of coincidence, the formula, and one more formula the formula for coefficient of coincidence that I told you is number of observed double crossovers by expected double crossovers. Because if you see the options, we have to calculate the double crossovers, single crossovers, right? So first of all, how we can calculate the observed or expected double crossovers? So the very first thing is that theoretically we can calculate the proportion of double recombinant gametes by using multiplication rule of probability, according to which the probability of two independent events occurring together is calculated by multiplying the probability of the independent events. So here if you see, this is the single crossover event which has occurred independently, and the crossing over between the E and SE allele has occurred independently. So these are two independent events. So we can calculate the expected frequency. So the 20 centimorgan, so we have to write down the probability of the single crossover event 1. This, let's say this is single crossover event 1 and this is single crossover event 2. So here it is 12, so 12 by 100, right? So you can write down 0.2 and 0.12, okay? So you don't have to write in centimorgan, you have to write in the, like we have to keep in mind that we have to write the values. So keeping in mind the probability in terms of that, right? So 20 by 100, so that gives you 0.2, and here it is 0.12, so that gives you 0.024. So this is the expected frequency of double crossover, and observed frequency of double crossovers will be, so again, you know the coefficient of coincidence value, which is 0.6, and we have observed double crossovers, which we have to find out, right? And expected double crossovers, just now we have calculated, that is 0.024. So don't you think we can calculate observed frequency of double crossover? So what you have to do is simply you have to take these value of expected, like you have to, for the observed double crossover, this is what you need to do, right? So on multiplying, you will get 0.0144. And even you have to keep in mind that the total progeny given is, like we have some total progeny given to us, right? So when you multiply it, the observed double crossovers, like for the total progeny mentioned, it will be 26. Then we have to calculate the single crossover events also, okay? I think please note, I think it will be total progeny here, it will be 1800, not 18,000, okay? So now to calculate the single crossover event, it will be like you have to keep in mind. So we know that the single crossover event probability is 0.2 and total progeny is 1800. So you need to keep in mind that when you calculate the distance between the genes, right, when you calculate the recombination frequency, we consider the double crossovers, right? If you remember just few minutes back, we solved the previous question. So number of recombinants, we have considered double crossovers also, right? But here specifically only we need single crossovers. So whatever double crossover we have calculated, progeny we have calculated, you have to subtract it from here. So 26 we have calculated. So when you multiply 1800 with 0.2 and whatever value you get, you have to subtract it from 26. So 334. And similarly, you have to calculate for single crossover event 2, that is when crossing over happens between the allele E and SE. So here you get 190. So total recombinants will be 334 plus 190, and also you need to include the double crossover, which is 26. That gives you 550. And non-recombinants will be, so you know that from the total progeny, if you remove the recombinants, you get the non-recombinants. And all these combinations, the values are given in option 1, and therefore answer here is option 1. So I can understand this is a little bit tricky question, but with practice, you all can easily solve. All you need to understand is the strategy of solving such questions, which are discussed in this Brahma series sessions. Now, quickly moving over to another question. Considering the following crosses involving gray and yellow body color, true breeding Drosophila. So here you can see this is the cross mentioned. And assume 200 F2 offsprings are produced in cross 2. Which one of the following outcomes is expected? So basically, you have to find out the outcome of the cross 2 in F2 progeny, F2 offsprings. So I hope you have learned something new today. We have discussed 2 questions which were a little bit tricky, and I'm sure that now you have understood that if such questions are mentioned, how you have to solve them. To quickly answer this question, considering the following crosses involving gray and yellow body color, true breeding Drosophila. So a table is given to you, cross 1, cross is mentioned, F1 progeny is mentioned, F2 progeny is mentioned, and even cross 2 is mentioned, and then F1 progeny, but you have to, is mentioned, but F2 you have to calculate. So what do you think is the Correct option here. Just look at the crosses carefully. Gray female and yellow male, and cross 2 is yellow female and gray male. So can you tell me what kind of cross it is? What kind of cross it is? Gray females with yellow male and yellow females with gray male. What do you think? What kind of cross it is? Have you heard of back cross, test cross, reciprocal cross? So what kind of cross it is? Initially, the female which we considered was wild type and male was with the yellow body color. Now you are considering in another cross female to be with the yellow body color and male to be with the gray, that is wild type. Yes, Surendra, you are absolutely right. It is reciprocal cross, right? So what you see over here is a reciprocal cross. So what you do in a reciprocal cross? Yes, Tejaswani, absolutely right. So F1 progeny is mentioned, F2 progeny, what it will be? Okay, Anupam, yes, it is reciprocal. So now just write down the genotype. Okay, the hint is write down the genotype of the yellow female and the gray male, right? And then you can again write down the genotype of F1 progeny. Based on that, you can determine the F2 progeny. So what is the answer here? You can see some values are given, some numbers are given that 97 will be gray male or 54 yellow female or 102 yellow male, 46 yellow female. In options, all four options, some values for separately mentioned for the progeny. So just write down the genotype. So it is mentioned that you have a gray and yellow. So gray is dominant, that is wild type. So you know wild type is most common, so it is dominant. And yellow is recessive. So yellow female crossed with gray male. So this will be, if you write down the cross 2, it will be yellow is a recessive trait. So let's say we are representing the dominant trait, that is gray, with the uppercase letter G, and the gray, that is the yellow, please correct, the yellow, which is a recessive trait with lowercase letter G. So yellow female, so we will be writing this genotype, and gray male. So this must be the genotype, right? So obviously, you know, if you're performing this kind of cross, what will happen? So here we'll have the gametes, and here we'll have one type of gametes. So either these two can come together or these two can come together, right? So either we'll get all males yellow and all females gray. So this is what we are getting, right? So which are the same I have mentioned here. Now, what if the F1 progeny, like how do you get F2 progeny? By like when on crossing the F1 generation, you get F2, right? Usually in Mendel's dihybrid cross, we have seen selfing of F1 generation will give you F2, right? So here you have all males, which is yellow, right? And they are crossed with the female, which is gray, right? So in such type of cross, what will happen? What do you think? What will be the gametes formed? So these two can come together, or this one can come together, right? So either you'll get this combination or you'll get this combination, or you can get this combination, and the fourth one is this combination, right? So basically, if you see here, so like this one, so you can see here, this one will be the gray female. So in heterozygous condition, we have the wild type allele, phenotype is observed, right? Then here it will be like we have recessive allele in homozygous, so yellow female. Here again we have the allele for gray body color, so gray male, and here you have the yellow male, right? So all these will be in what proportion? Equal. All of them will be in equal proportion, that is 25% each. Now, if you look at the options, here, if you see the ratio, like here it is 97, 54, 49, no. You have to look for the option where you are getting equal proportions for all. So 200 is the total progeny, so that means we should get 50, 50, 50, 50, right? All for each 50, right? So here if you see it is 52, here it is 49, 48 and 51. So approximately it is like 50, we have 50, right? So this is the reason answer here will be option 3. Yes, Anupam, it will be option 3. So moving over to another question. So we have, we have 3 minutes. So let's solve this question quickly. So this will be the last question for today's session. So mutations affecting the direction of shell coiling in the snail Limnia perigra are known to exhibit maternal effects. That is, the phenotypes of progeny are determined by genotype of the mother. An F1 snail produced by a cross between two individuals has a shell with right hand twist, dextral coiling, and this snail produces only left hand sinistral progeny on selfing. If the D, that is this allele, represents dominant dextral allele, and this allele represents recessive sinistral allele, what is the genotype of the F1 snail? And options are given to you. So I'm sure by looking at the question, now you have understood that this question is based on maternal effect. And you have to recall the what happens in F1 generation, what happens in F2 and F3 generation. So quickly answer this question. Okay, Anupam, you are saying option 3. Anusha, Aishwarya, very good. Option 3 it is. Okay. Surendra, okay, you are also saying option 3. So what is maternal effect? You have to recall here what is maternal effect. So maternal effect, I'm sure you all know it's a condition where the phenotype of an organism is always determined by the genotype of its mother. And the very popular example associated with this particular maternal effect is of Limnia perigra, which is a freshwater snail. And it is observed that the shell of the snail of this snail can be either clockwise or dextral, or anti-clockwise or sinistral. Now, as mentioned in the question, that this particular allele is for dextral and this particular allele is for sinistral, right? So F1 generation genotype can be dextral or sinistral. It will depend upon genotype of female parent. So for example, in this A figure, if you see the mother is dextral, having dextral coiling, so you can see the genotype of the female, homozygous condition is there for dextral allele, and father or the male is sinistral. So here you can see that the male is having the recessive allele, the sinistral allele in homozygous condition. So obviously you know that in such type of cross, we get the F1 generation, which is heterozygous. Now, if you perform a reciprocal cross where now the female is sinistral, having sinistral coiling, genotype you can see for sinistral coiling, and the male is having the genotype for dextral coiling. So again, F1 generation will be heterozygous. There is no change, but you can see the coiling here is sinistral, right? Now, in F2 generation, if you perform selfing of F1 generation, the ratio you'll get 1:2:1 in F2 generation, right? But you can see all here are dextral. Even this particular coil, is this particular progeny, if you see having the genotype for the sinistral, it is showing dextral. The reason being is that here the genotype, the phenotype of the organism is determined by the genotype, which is here heterozygous, right? So therefore, the even this particular progeny with this homozygous recessive allele for sinistral can be is dextral, as genotype of the female parent carries dominant allele. So here in the question, it is mentioned that F1 snail has dextral coiling, and on selfing, the snail produce only sinistral progeny. So because here the progeny is sinistral, so that means as the phenotype of the progeny is determined by genotype of the mother, the female parent, so parent definitely must be having the sinistral allele in homozygous condition. So therefore, answer here is option 3. So I'm glad to see the response. All of you have answered it correctly. So I'm stopping here, and this is all for now. So please practicing, and more you practice, more you learn, and I'm sure these sessions are really helpful for you all. And in case of any doubt, you know we are always here to help you. So all the very best for your with the preparation and for solving the questions. So did you like the session? Did you find it useful? Did you learn something new today? Please quickly let me know in the chat. Okay, so thank you everybody for attending the session. We'll see you in the next session. So take care and